everybody, today we're learning all about the National Peanut Research Lab. So join us as we explore how they're helping create a better peanut for producers and the future of agriculture. Hey everybody, we're here at the National Peanut Research Lab in Dawson, Georgia, and we're talking to Dr. Marshall Lamb. So Marshall, tell us a little bit about who you are and where we are today.
Well, we're here at the National Peanut Lab, as you said. We are one of the only labs in the U.S. that's solely dedicated to peanut research in the United States. We have scientists doing a wide range of peanut research from initial variety development all the way through processing and product development.
Okay, well, can you show us a little bit about what's going on here at the lab and all the research you're doing?
I would love to show you.
All right, let's go.
Okay. Megan, this is Erica. Erica, this is Megan.
Erica.
Erica is doing a lot of really innovative research on peanuts and peanut roots in the effort to help us develop peanuts that will become more water use efficient.
Okay, so Erica, tell us a little bit more about what you're doing, what we're seeing here.
Sure, so I predominantly study how plants are using water, and the methods are really easy to do and really established in leaves and stems, but not so much in root systems. So this year we started with a new kind of easy experiment where we thought, is there a way that we can isolate these root systems? Because right now the kind of standard method is shovelomics, and that's just going out in the field with a shovel and digging. And you don't know if you're chopping off these roots, which plant you're getting. So we just created these plywood boxes, stuck some nails into them, and we stick one peanut plant in. We let them grow happy and healthy throughout the season, and then after about 130 days, we open up these boxes to get something like this that you're standing over there. Which is kind of a fully intact representative root profile. We're able to slowly wash away the soil, and after an hour or so it looks like this. We take high quality images of these root systems, and then with AI and some machine learning we're able to kind of pull some hard data, some numbers out of that tells us about rooting depth, rooting structure, and we can kind of extrapolate that to different varieties and different soil types and different environmental scenarios.
Okay. So what's your overall goal for the research?
The overall goal, I would say, is we want to see how different varieties are using water differently in different soil types in response to different heat stress, drought stress, stuff like that.
Okay, so do you stress these out at all, or?
These ones have not been stressed
normal?
out. These are Georgia 06G, which is the standard kind of peanut that we use in the region here. And this is just kind of baseline information we're getting does this method work? And now after 2 years of doing it, we can say, yes, this method works. And so now we can start looking at, like I said, different soil types, different stresses, stuff like that.
Okay, so how far down do the roots typically go? That's really long.
It is really longer than we expected, but they're also, like I said, well watered and healthy. You can follow the tap root down, so this is this main root, and it goes all the way down about 6 or 7 feet. So that may vary, like I said, depending on soil type. Sometimes there's hard pans that develop in different soil horizons. So there's a lot of new things that we can look at now that we have this method established.
Megan, this is Dr. Ron Sorensen.
Hey.
Ron is the lead scientist on our crop production unit and focuses on irrigation strategies for growers.
Okay, cool. Well, Ron, tell us about what you've got going on here.
Well, this is a little different than kind of what we've talked about with irrigation, but we're actually looking at ways that we can stop or reduce the possibility of aflatoxin coming into our peanuts. Years ago, a researcher here by the name of Joe Dorner came up with Afligard, and that is a Aspergillus flavus that has non-toxigenic strains that will not produce aflatoxin. And so he found that, and we're using that, and we put it on barley hulls or things like that in order to put it out on the field to reduce aflatoxin. we talked to a colleague of ours and said, well, can you take that aspergillus and put it on seed? He said, well, sure, why not? So we tried it. It's not registered to go on seed at this time, but we thought we'd at least try it and see if we could find a way using agronomics to stop aflatoxin or to reduce the chance of aflatoxin in the field. And so he coated some seed for us, and we've actually got them planted here. And then we took aflatoxin or Afligard that they normally use and spread them out, and that's at 60 days after planting. So in this plot we have Afligard on the seed, we have Afligard that we put on 60 days after, and then we have a non-treated, so we can compare that. And then also behind that we have irrigated, and then we droughted these at 90 days so that we can see if we can get aflatoxin to come in and aspergillus start to grow. We know that aspergillus starts to grow at high soil temperatures and drought. And so that's why we got the drought going on here. These are controlled so that when rain comes, the sheds will automatically close, and then after so long a period, if there's no more rain, they'll automatically open. So these aren't closed all the time, and we don't have to have someone here pushing a button.
Okay, pretty cool. So what do the toxins, what you're talking about, what do they do to the peanut plant?
The aflatoxin is a toxin, and so in consumers, we can't have more than 10 parts per billion, and at least down to 4 parts per billion, and that's a very small amount. And so we could just have 1 or 2 seeds that may contaminate the whole load. And so if we can reduce the amount of aspergillus actually growing and excluding excluding the toxigenic strain with the non-toxigenic strain, then hopefully that we wouldn't have aflatoxin coming into the peanuts. Because that's if aflatoxin does occur in the peanuts, the price of the peanut goes down drastically for the grower, and so we don't want that coming into the fields and into the crop. So if we can reduce that any way we can through agronomics, that's what we want to try and do.
How would the producer know if it's in the field in the peanut?
They have to do it chemically. And also when you take your peanuts in to be graded, they actually will crack a peanut open, and if they see the fungus, which is a green color if I remember correctly, if they see that, then it automatically goes what they call SEG3, segregation 3, and it has to go to oil stock. And so the price goes from $560, $550 down to like $130, $140 a ton. And so it goes down quite drastically just by having aflatoxin present. That's why it's such a big problem.
Okay, and so you're trying to see if you can coat it on the seed,
That's
if there's
right.
a difference, or plant it 60 days
That's right.
out, or not plant it.
The 60 days after is putting that Afligard on. Now what that does is it makes it so that the farmer has to come and put out an extra trip across his field to put out the Afligard. If you can put it on with the seed, it goes on with the seed, and you're already going through the field anyway. So that's going to save him that amount of money, not having to come across the field again.
But from a consumer standpoint, the industry spends millions and millions of dollars every year to ensure that aflatoxin is never introduced into the edible food supply. And they have sophisticated technology that can segregate the peanuts, and so consumer safety is always first. And the peanut industry is very aggressive and very good at maintaining that. This is a way that we can cut cost out of the system. It would help growers by not having the segregation 3. It would help our processors by not having to process as many peanuts out if we're able to deliver them to the buying stations clean to begin with. But consumer safety is always guaranteed because the shellers stringently test peanuts before they go to manufacturers to ensure that aflatoxin is not present. Megan, this is Dr. Phot Dang. Phot's one of our research scientists here, and this is Dr. Joseph McIntyre, who is a research ag engineer at our lab.
Okay, hi. So tell us a little bit about what you're doing here, what the research that you're doing.
I'm interested in drought research, and so what we'd like to do is to identify drought lines so we can use it as parents for crossing. We have collaboration with Dr. Charles Chin at Auburn University, and also with Dr. Alvaro, who's a physiologist who has his groups come in and help us with measurements of the plants.
Okay, cool. And then what do you get to do, Joseph?
Oh, I'm here in this particular project supporting the research. We're developing a new way to essentially observe the peanuts long term using time-lapse cameras. They don't look much, but this little camera will let us essentially watch the peanuts all day, and well, if we put up the lights, all night, to see what they do by photographing them at about 1 minute intervals. And it turns out the peanuts dance at that rate. And we can see them wilt in the sun, recover with the dawn, and it's, well, essentially we've set it up so using the arms, we get like a drone that never lands, all for the strategic use of 2x4s.
Pretty cool. So what specifically are you doing with your research?
This particular experiment is, we are evaluating 6 different lines. The ones that we have put in here are already evaluated in the fields in the past years, and we identified 2 particular mechanisms of drought tolerance. One we called a drought saver, which means that the plant has the capacity to recognize stress early and reduce its photosynthesis and other gas exchange type of physiological measurements. And also we've noticed that a little bit of a reduction in canopy size as well. But that's one mechanism. The other mechanism is a water spender, which we would continue to see these plants, once it recognizes stress, it's continue to do photosynthetic rates that are relatively high and gas exchange, and max out its capacity to use that water. So the 2 different mechanisms really don't have a yield loss component at this point, but I just want to justify that this area, because it's a southeast, it has very high humidity during the evenings, but as the heat and drought occurs, then the humidity drops, and then plant stress really, that's when the separation occurs. So that's what we're trying to evaluate. So for this particular experiment, we have 6 different lines here, and there are 2 rows representing one particular line, and each block has 1 replication. So comparing the different lines in the same treatment, we like to see that what are the differences between some of these lines, if they're separating based on susceptibility or their mechanisms of drought tolerance. Comparing that control to the ones in the back where we irrigate that, and 2 other plots that are similar in this particular experiment.
Okay. So you're taking a variety that maybe they're wanting to put out, and you're making sure that it does what it says it's going to do?
The key is to really test its parents, just to make sure that if we can use these parents to, as cross potentials, to see if the particular genetics or its production capacity could be transferred to siblings, and eventually have variety releases in through our programs. And again, the joint breeding program that we have with Auburn University is very helpful, because we split the work in very, very subjective areas where we can really critically look at different treatments and quality traits, and then supplement that to make sure that the field evaluation is very well tested at the same time. And so once the varieties or the potential variety is good for release, then that's what we hope to help with that breeding program.
Oh, okay. That's cool.
Megan, this is Dr. Paola Faustinelli. Paola is our newest scientist here at the lab, and she is a research plant physiologist.
Okay. So tell us a little bit about what you do, what your work is, and your role with peanuts is.
Okay, well, here I'm working with in vitro peanut plants, so peanut in vitro culture. So I'm using organogenesis, that is a process of formation of organs from different type of explants. So you can use, for example, cotyledon, leaf, or roots to induce shoots. And that is possible because every single cell has the whole information, so the DNA, to reproduce a complete and fertile plant. So first of all, you have to disinfect the seeds, and then from there you can extract the explants that you are going to put in these Petri dishes, and these contain media, which sucrose, and different type of growth regulators, then they will induce shoots. And then when you obtain the shoots, you transfer to these magenta boxes, and for elongation and rooting. And I can also induce flowers, and if you can see, there is a small flower over there, and also pegs under the same conditions. So for example, here you have a peg, and a small peg. So why is this important? Because every protocol you have to adjust for the cultivar that you're going to work with, and it's important for my next objective, that is work with CRISPR. So CRISPR is a gene editing tool that has been used to improve crop quality. So for example, for peanut growers, it's really important because, for example, they are dealing with a leaf spot that is a disease, that costs a lot of money. So what can we do is, breeders are trying to find, like Dr. Massa is trying to find a gene, that confer resistant to that disease. So she's checking plants, trying to find a resistant, and then she will study the DNA, trying to find a mutation. And then she will start doing, crossing between the resistant and the susceptible. And after between 10 and 12 years, she will obtain a new cultivar, right, resistant to that disease. So what can I do with CRISPR? So with CRISPR, you can transfer that mutation directly to the cultivated peanut. So, and that will take me maybe between 5 and 7 years. So it's half the way. But the only problem here is that what I have to do is edit the DNA of one single cell, and then from that single cell, I have to induce a shoot. And that's very simple to say, but it's very difficult to do it. So that's what I'm trying to do here.
So you're kind of looking at cutting that whole breeding time in half by extracting that one cell or.
Exactly.
Oh, okay, that's cool.
Good morning. This morning we're out here at one of our off-site research farms where we're doing a lot of work on aflatoxin in peanuts. One of the tests that we have multiple tests, but one of the ones we're focusing on the most is the use of Afligard as a biocompetitive agent to reduce aflatoxin in peanuts.
This research that we're doing here entails a lot of agronomic type work. We have 3 irrigation regimes. Behind me, back over here, we actually have a dryland regime. And then where we're standing right here is actually irrigated up to 90 days and then droughted after that. And then on the other side of us here, we have a full irrigation. And that allows us to look at the agronomic techniques that we're trying over different irrigation regimes. The irrigation regime at 90 days, we're looking at a possible, if a farmer is going through the year and his pivot breaks down and he can't get it repaired, if he starts to get into a dryland situation or a drought situation, what can he do to possibly save that crop to not lower the risk of having aflatoxin? Also, one way that aflatoxin can come into a field is if it's hot and dry at this stage of the field. We're almost ready to harvest. And so if we get a drought now, the possibility of aflatoxin and the risk increases. And so if we could keep soil moisture in the soil longer, then that would mean that aflatoxin or the fungi would not sporulate and grow. And so what we're doing here and what you see right here is we actually cut the plant. What that does is that stops the transpiration and it leaves the soil moisture or the water in the soil, and hopefully our theory is that Afligard or aflatoxin will not grow in the soil because it's wetter. The last few years when we've done this, we've actually have seen anywhere between 3 and 5 days that we can save moisture for that period of time compared to one that's fully or normal without being cut. So if we can save 5 days and we look like we might get a rain, we may have saved the crop. Now, if we cut, we also know that from previous research that we lose about 12% of the yield. And so the risk comes in is, do I lose 12% of the yield and not get aflatoxin, or do I go ahead and let it grow, possibly get aflatoxin, and lose that crop to a segregation 3, which is a lower yield at a lower cost or price per tonnage. And so that's the risk that the farmer is going to have to take. And that's where our economist comes in, and Dr. Lamb, and saying, here's the risks, here's the change, here's the yields, what can we do? Is this a viable option?
So the peanut's still under the ground?
Yes, the peanuts are still here. These have been droughted, and so they don't look quite as good because of the drought, and we cut the leaves off, and so they can't transpire and really can't grow. And so these do look really bad, but there are nuts under here. They are viable. Probably when we cut them at about 90, 95 days, the crop was already there, and so whatever was there probably went to fruition, and they're probably good nuts. They're just not as many as compared to here.
Oh yeah, because you're just leaving it
right.
on to
And so we
continue
will look at we will actually shell the peanut and find out what aflatoxin may be in this peanut versus this peanut versus the one that's in the irrigated versus the one that's in the dry land. And that way we can compare all of them and say, is this a viable option? Should we go out and cut? Should we go out and do something else? You know, is there some other option that we've got? And that's what we're looking at. The Afligard on the seed, the Afligard put out at 60 days, different techniques that we're trying. Another technique that we're actually trying is that we know that Aspergillus flavus is a fungus which actually is here to decompose organic matter. So if we can kind of decoy them and go on to some other organic matter instead of the peanut, maybe we can get them over here and not on the peanut. And so what we did is at the first of the year, we actually put out 750 pounds per acre of peanut hulls and put them in certain plots out through here and say, you know, and kind of use that as a way to decoy them away from the real peanut if we do get the situations of where they're, you know, it's dry and whatever, you know, dry and hot. Are they already over here chewing on these and trying to decompose this peanut instead of our real peanuts that are growing? So that's a test that we're looking at. It's a pie in the sky type thing, but who knows? You know, something like that might work. And it would give a way for the shellers to get rid of their peanut hulls if it works. They could get rid of recycle and repurpose those peanut hulls.
Okay, you just lay them on the ground?
We just laid them on the ground and then we slowly tilled them in.
Okay.
So they are tilled in, but they're only tilled in just a little bit, about where the root zone is, or the peanut pod zone. That way we can look, and every year we come in and we actually take samples of the soil and have it tested to see if we actually do have Aspergillus flavus, and if the Aspergillus flavus is the kind that will actually promote aflatoxin. So we can look at the different types.
Yeah.
So Megan, this is the peanut profile board or maturity profile board. This technology was developed a long time ago in order to help growers understand when they need to dig their peanuts. It was developed by the Agricultural Research Service, which we're with, the University of Georgia. The work was funded by the Georgia Peanut Commission, and it was really successful work because growers never really knew when to dig based on the peanut maturity. And one of the factors is the fact that peanuts are an indeterminate fruiting crop, which means throughout the entire life of a peanut, they will continue to flower and continue to put down new pegs, which ultimately can become new peanuts. So you have varying maturities on this board, going from the white class, which is the least mature, very, very young peanuts, if you will, on the plant, going across to the different darkening colors. And as you can see, the colors really start changing. And when we get to this point here, in the black-brown category, that is where the peanuts are maturing, developing their flavor and such like that. And generally, we come and when we have a good slope on the peanuts and we have a sufficient amount within 3 days, the recommendation is getting really close for a grower to go dig that field.
Oh, so 3 days to from the end to start digging it.
And growers normally will go grab a sample of peanuts and then they'll take it to the cooperative extension agent in the county because they are so important for growers. I can't underestimate how important extension service is at the county level and actually the state level. But then they'll blast them and help the grower board these and say, all right, you're 14 days out from planting, but in this particular case, we look like we're ready, or from digging, I mean, and this one shows that we're right up on the edge of digging, so this field will be dug and harvested very soon.
Okay, so they help them make that plan of action of
Yes.
what to do next. Okay, that's pretty cool. Is this the time now to ask about the economic importance of peanuts and like where they land, and the impact?
Peanuts and cotton are generally the most important crops we grow in the Southeast as far as row crops. Economically, peanuts are probably the #1 crop because not only do we grow peanuts in the Southeast and other areas, of course, but we also process the peanuts through our shelling plants in the Southeast, and then from there we'll ship them to manufacturers to become peanut butter, candies, and such like that. So the economic impact of the peanut industry in South Georgia, Southwest Alabama, Northwest Florida is extremely important to the job base and the overall economic activity in the region.
Okay. So what are the career opportunities for students that maybe, they can't be on the farm, so how can they help the peanut industry?
Well, actually they can be on the farm. We hire students in the summertime, mainly college students, and we give them the opportunity to come out and not just look at or read about the opportunities in the agricultural field. They will be out here helping us install sensors, monitor sensors out here when we're planting, and it's just so rewarding to have a student that comes out that's interested in agriculture, and we're able to work with them over a summer, and then they decide that that's going to be their career path. But agriculture is not just farming. It is so high-tech these days. It's computer science, it's machine learning. It is so much that goes into decision making today that we did not have 5, 10, and especially 20 years ago, because it's all high technology that we use today. And growers on a production side are all using GPS guidance. They're all using pesticide applicators that are guided by computer systems. And then fertility wise, we've got zone management that growers are using. And that all lends to helping the growers yield a higher net return. But again, back to sustainability, it helps with our continuing improvements in sustainability.
There are a lot of opportunities. It's really cool.
And like I said, we have students that come down. We had one young man at our lab that came to work for us. He's very good at computers, but yet he worked with Ron and Erica on the crop side, and now he's going back to graduate school to combine those two into a program, graduate program. So we're really excited when we can instill the knowledge of agriculture into the students, but then to inspire them to go further in their educations and be an integral part of the whole industry.
Yeah. And then, like I said, they find out about the jobs the internships and positions through USA Jobs, right?
Right.
Okay. Cool. Well, thank you all for showing us around.
Thank you.